Welcome to the Biotech Startups podcast by Exceeder. Join us as we speak with first-time founders, serial entrepreneurs, and experienced investors about the challenges and triumphs of running a biotech startup, from pre-seed to IPO, with your host, John Chi. In our last episode, we spoke with Mark Kotter about growing up as a curious child in Canada, his exposure to a broad range of cultures and perspectives moving all around the world, and how his dyslexia led him to focus on science and mathematics at an early age. We also touched on his experience as a researcher and PhD student at Cambridge, where he transitioned from medicine to synthetic biology. If you missed it, be sure to go back and give part 1 a listen. We continue our conversation in part 2, discussing Mark's transition from his PhD studies at Cambridge to the Medical University of Vienna, where he became a resident in neurosurgery and ran his own research group. We also touch on his experience at the Max Planck Institute for Experimental Medicine, his return to Cambridge, combining clinical practice with research, and his shift from academia to entrepreneurship, which led to the founding of his first biotech startup.
So I was a little bit homesick, and I thought, okay, after my PhD, why don't we try it out? Homesick is weird because my definition of home now is where my wife is, a locality. But, there was a certain sense of that. So there was the effort to create and fund some spinal cord injury research in Vienna. And I just happened to have that interest. So during my PhD, it became clear that I wanted to do neurosurgery and that I wanted to, go back to spinal cord injury just at the beginning. My house of the years, I spent really more in ophthalmology and psychiatry and maybe a bit of neurology. So the surgical part came through rather late, to be honest. So then I joined completely serendipitously. There was an opportunity, and I was able to set up my lab. I had some core funding provided, and it was amazing because everything came together. I had 2 PhD students. I kitted out my own lab, and I started training. But then, relatively in a short period of time, I enjoyed the freedom I had in the lab, no supervisor, but being able to run a group. On the medical side, it was incredibly hierarchical. I mean, like probably US in the 19th century, and I just couldn't stand it. It threw me off, and then I decided to see whether there's another way, another place where I could combine research and medical training. So I got this chance to work in the Max Planck Institute in Göttingen and join the medical faculty there. And what I learned is even worse there. Not the Max Planck Institute was amazing, but by then we published our first paper, and people in Cambridge took notice of me again, and then I got invited to apply for a junior faculty position. So that was a really unusual position. It meant that you could start your own lab, but you would still be in medical training. And that is a lot of sort of headache, as well. Brings out a lot of headache. But it was, a tremendous opportunity, and I jumped at it. And then I moved my little family. My daughter was maybe not quite a year old or so, and we went and relocated to Cambridge, and the rest is history. So I only once escaped from Cambridge.
For not that long. Yeah. I'm feeling homesick. And then I guess I'm back.
Exactly.
And I guess I have a quick question. So just go back to the first, research group you ran right after your PhD. Can you talk a little bit about, you said no supervisor, and now you have 2 PhDs.
Yeah.
what was that feeling for you, like to run your own, run your own lab? Because like
I
that's
mean, it's
markedly different.
exhilarating. It's exhilarating, but at the same time, I was pretty scared because we had to put everything in place, and they were real PhD students. So, I had to teach them everything,
Yep.
from how you do the cultures, to how you pipette, to, and we knocked it out of the park. I mean, we published a paper in Brain which was a very, for an unknown person, a very good first pass. And so, no, it was really, it was scary at the beginning, but then as things came together, you get drawn in, and then, things just started to work out, and yeah, it's amazing actually. Yeah, I mean,
And
incredible
I
that I had the opportunity directly
and
after my.
I love the dichotomy as well, where everything you're describing, I can see the energy. Like I can see the
Right.
energy of like running a lab. And then we think about you go to like the kind of the residency medical part where it's just like the hierarchy. You're like, oh God, like this is like a a night and day experience.
Yeah, it was totally, yes, yeah,
And so now, you're running your research group, you're having this night and day experience, and now you got the invitation to go back to Cambridge. Can you
yeah.
talk a little bit about, relocating your family and starting that, that role while you're still doing your training? And you said there's a lot of headaches. Can you describe, what, kind of what went down?
So, I'm super lucky with my wife. She just said, "Yes, let's go," and that was it. So there wasn't, you know, any palaver around that. And um, the the lab that I joined, so the institute was set up by Roger Peterson, one of the sort of founders, I would say, of human stem cell biology. And it was a very special place. It only existed for maybe 7, 8 years, and then the university shut it down. But it was an incredibly special place because we were like an outpost. We were, a handful of PIs with their groups, and everyone was interacting and working together. When everything at that point in time was really difficult to do with human stem cells. We didn't have the media yet. So, we had to mix our own media. And for months, the media wasn't quite right and you lost all your cultures. Or many of the techniques that we now use were just introduced. CRISPR, for example. So it was quite an extraordinary time. And Roger, again, was very thoughtful how he combined different labs. And we had some mathematicians there as well. So it was quite a special environment, very open. So information flew very easily across the groups, and that lifted us all up. When I look at my fellow PIs, all of them were extremely successful. Whether they took on very senior academic positions in Cambridge or elsewhere, but they were all, very, I mean, they're, I have a ton of respect for them. Amazing, actually.
Absolutely. And I mean, it sounds like, you're taking a leap of faith, but it was just like a slam dunk, like more than anything. Cause like, and also one thing I love hearing is, and something that's I always like highlighting too, is how important, having the support of your significant other and your family is in all of this. Like no one
Yeah.
could do it alone. I talk with my wife and I think her all the time about, for Exceeder, just like in the earliest days, just like when when it really did feel like a leap of faith, that she didn't stop believing and said, yes, let's go for it. And I can imagine, that's relocating the whole family that's not, trivial. It's very much
No.
it's a big move, and so I think that's critically important in anyone's career and journey, is this, having the support.
Having that support is absolutely critical. Yes, I mean, yeah, I'm very lucky in that regard. But back to Cambridge, there also were two worlds. So you had the research world, and I probably was in one of the best environments I could, dream of. And then I had the clinical world, and it was very different. So the good news was it was very meritocratic, but still there was a huge reluctance of some of the old-fashioned neurosurgeons that their trainees should, spend time away from the bedside on the bench. It was nearly an insult to them, because at that point in time you were still, a glorified slave, and so that was also, I think, a very tough environment. And I think one of the character traits I have is I just walk through walls if I want something, without hopefully breaking too much. So I would say I was broken in by the system to become the neurosurgeons that they wanted to see, but I also broke the system.
Yeah.
And actually, now Cambridge is one of the premier places, probably in Europe, where clinician scientists in neurosurgery can pursue their research interests.
That's amazing. And I didn't realize how much of a kind of like juxtaposition the kind of like the clinical side and the research side. I've never,
Yeah.
been in the clinical side. So I this is the first time I'm actually hearing about it. But I love
Yeah.
that now it's kind of like, it's an embrace of, rather than just like these
Now
things that are
it is
at
much
odds.
more sort of come together, but at that moment in time when I trained, it was just like absurd that you would even combine those or think or seek to combine those two aspects. And in theory, it actually makes a lot of sense.
Yeah.
I mean, why wouldn't you? Is
Yeah.
my question now. But look, one of the things that happened, of course, is that, and this could have only happened in that context. On the basic science side, I was exposed to the newest technology around stem cell, pluripotent stem cell research. And then on the clinical side, I had access to patient samples. And that combination was pretty amazing, because what I learned, and that was another big learning opportunity, was that the cells that I'm most interested in at that moment in time, oligodendrocyte precursor cells. And we were using mainly rat and mouse models, and we didn't care so much about human cells because we thought they're similar anyways. And there was a protocol that was published to turn pluripotent stem cells into OPCs, and it took something like 170 days. So imagine me telling a PhD student, you've got half a year to differentiate your cells, and at the end, you're not sure whether you have some or not. But at that point in time, you can do an experiment.
Yeah.
So
I'm sure they loved hearing that.
Okay, so that wasn't very enticing. But then what happened was we got some brain, samples, and we extracted some these OPCs, and they looked in culture very similar to the rat and the mouse OPCs. But they behaved completely different. They didn't differentiate the way that the mouse or the rat OPCs did. In fact, some of the growth factors that had the opposite effects. And I remember, realizing this and thinking someone just, the floor beneath my feet is just evaporated. I'm in free fall. Because I said, okay, I'm doing all this research on rats and mice, and all that I can ever dream of is become a rat doctor or a mouse doctor.
Yeah.
So that really, and then the complexity around, the differentiation protocols, which were untenable. And that really was what drove me to look into different or alternative scenarios. And this is when I learned about, cell programming, essentially. So Yamanaka, of course, the foundational program that everyone uses, but actually you can trace it back to the 1980s. There's a chap called Harold Weintraub who showed that a transcription factor can convert fibroblasts into muscle cells. And of course, around 2010, when I started in Cambridge, Marius Wernig, Tom Suthoff, they published their direct reprogramming protocols, turning fibroblasts into neurons. And then they did another one where they turned liver cells into neurons. And then they applied the same paradigm on pluripotent stem cells. And that paradigm questions everything that you think you know about biology. So the dogma at that moment in time, of course, influenced by development biology, was that cells are defined by the history. And it's an epigenetic state. And, we talk about Waddington's landscape. But actually, that data is in total contradiction to that. And that was noted by Weintraub, who said, well, maybe cell identity isn't that fixed. Maybe we should call it metastable. And then he said, and these cells don't undergo any of the developmental steps, as far as I can see. It looks like this transcription factor that I've identified kickstarts a feed-forward loop, and somehow that becomes self-maintaining. Of course, now we call these gene regulatory networks. And then the third question he asked was, can we combine cell identities? Can we create hybrid cell types? So he showed that that's possible as well in certain circumstances. And then he died from a glioblastoma. And all this knowledge gets lost. So how did we get there? I mean, it's essentially having to rethink biology because of the constraints that the current thinking or paradigm, puts in place, and finding ways of sort of rethinking it, moving to a different place. That's a bit my history, isn't it? Moving to a different place,
Yeah. And also, what I'm seeing too, in addition to like a new environment, is like there, you have the clinical side, or, if we're thinking about back in the day when there used to be like church and state separate, but kind of combining two, disparate fields. Because like you wouldn't have had this like the floor underneath you kind of disappearing kind of moment unless you tried to like force these like, hey, like these mice, the cells that we're looking at in mice is vastly different than what we're seeing on the other side. But if you hadn't run through that wall to try and combine the worlds, perhaps you would have still been in this, like,
I'm sure I would have thought about it, of course.
Yeah, you would have just been focused on that.
Yeah. And then you see everyone is struggling with this. And actually, that's probably one of the main causes for what we call the translation gap. What works in a mouse doesn't work in a human being. Yeah, it's a different species. And most of the conditions that we try to treat are purely human. So just thinking about how we approach, I'm deviating now, but how we approach drug development, okay, let's find a cure for Alzheimer's. So let's start with mice. Oh, mice don't get Alzheimer's. In fact, there's not a single mouse on this planet with Alzheimer's. So what do? We engineer something into the mouse, a mutation, that then creates a disease that looks a bit like Alzheimer's. Now we have a model. Now we find a drug, a mechanism, and we can treat it. But if we then translate what we have found into the clinic, more often than not, we learn that it's a different disease or a different species. So how do you fix that? It's only by actually really using human cells from the onset onwards. And that's really one of the big motivations. The other big motivation for me was always to go back and find a way of improving spinal cord injuries. And I really thought cell therapies were the way to go, because it's so broken that you need to somehow, replace things. Things are lost, cells get lost and damaged. You need to put them back in because the body can't create them. So this was the combined interest in stem cell biology, and the barriers then pivoted me into what I would call synthetic biology.
Amazing. And I mean, it's again just like the timing of it all, and just kind of the kind of your ability to like join, like put together, like it reminds me of your kind of like early PhD experience, where you said we had mathematicians like in the lab, and we had various
Yeah.
disciplines, and it's very collaborative. And I can definitely see this through line where it's like we're only going to get better by people coming
Yeah.
up from different disciplines, coming together and really working together on a joint problem.
Yeah.
And as you were kind of first you come in as kind of into Cambridge first, as still trying to wrap up your clinical side, and you have your running, obviously you're running a lab. Can you talk a little bit about the progression as you started to, and I'm going to imagine you also had to teach. Can you
Yeah.
talk a little bit about that kind of progression at Cambridge, kind of up till now? Kind of what has that experience been for you?
A little steep.
That is, I can imagine. Yeah.
So look, you undergo neurosurgery training, and that's very time consuming, and it's full on. It's it's probably the worst thing you want to do if you want to do a research career.
Yeah.
So you need to be bloody hard nosed, and or driven. So, but it is what I felt was right for me, and so I took all these things, and and I hit through a number of, I think, glass ceilings. Over time, of course, you get used to it, others get used to what you're doing, and things become a little bit easier. And at some point in time, they actually recognize that maybe there's something in it. Maybe it's actually quite good for a department to facilitate this. So then, at the end of my training, I go away for a year to Toronto, which is the premier place for neurosurgeon clinician scientists. And that really again transforms, my thinking, and I'm learning completely new aspects that I didn't know much about. So very much I had a basic science lab, and I was very disconnected from the clinical, from my clinical practice. And in Toronto, I learned translation and clinical trials. So I came back. Before I went to Toronto, we discovered this OptioX technology. This is now the basis of my startups. Coming back as a consultant, your life changes again. Suddenly you're self-determined, and you can control your schedule. And if you can do that, again, you have much more flexibility to do other things. And this is then also the period when I started these companies. So in some ways, I was filling a void. I was sort of, it was so full on, and suddenly life became quite quiet in comparison. So it filled up very rapidly again.
Interesting. And so it was again kind of like Australia, but now you're in Toronto, and you had this kind of like
Yeah.
shock to the system, but just like fresh perspective in Toronto. Can we double click on like you mentioned consultancy?
Right. The consultant is staff. If you are fully trained.
Yep.
attendee would be, I think, the US expression.
Got it. And so you're starting to think about the startup world and the entrepreneurship world.
Right.
I can imagine very different than academia and clinical work.
Oh yeah. So I can spool back a few years actually. When I joined Cambridge, I said I'd be incredibly happy to run a lab and be a clinician. And I knew that there's some certain constraints around, salary in Cambridge. It's not as attractive, let's say, as it is in the US. And so I said, how can we fix that? Because I quite like it here. So my wife was an architect and I started a little business. And that, became, quite successful. And after a few years, we felt, it's fine. It'll help us, get by. I don't have to be worried about pension and things like that. That's covered. So, but it exposed me a lot to, dealing with banks, doing things that I never did before. And I quite like learning the new stuff and the rules of that game. And so, but I didn't want, once we found this Op2X technology, maybe something we also need to discuss, I didn't feel the need of spinning out companies. It wasn't the primary aim, quite frankly. It was a result of nobody seeing what this technology could do that made me say, okay, fuck it, I'll do it myself.
Yep, I'm very fascinated here. So, it seems like, these are realities. We have, needs bills we need to pay, and, school we need to pay for. And correct me if I'm wrong, was this like a family business? Like before all the startups, a family business that you and your wife started?
Yeah, it's completely different. She's an architect, so it's real estate. But it was a really good training ground to understand, financial models, how banks think about things, how you need to think about things. Cash flow plays an important role. I mean, all the basics are, applicable. So I felt I learned a lot doing that.
And honestly, that I love that, because I think now, just quickly doing a fast forward to 2024,
Yeah.
like in startup land, particularly in venture-backed startup land, we tend to forget exactly what you're kind of describing about, like, the fundamentals. Like, there was a world before venture capital existed where it was only the banks that were gonna fund you, or you're doing this out of pocket, and it's like a family business. And I can imagine you almost had like a crash course MBA, like but probably more intense, because it's like it's your family business.
You're on the hook for sure. Yeah, exactly. Yeah, some aspects, of course. I mean, the other aspects, which are probably the main thing now in my stage or company stage, is really how dealing with people. Bit.bio is now 200 people, so you can imagine that it's quite a complex environment that is very deep in science, but you need to make sure that everyone is in sync. So and it has ups and downs, like any sort of structure or organism. So I guess what I'm trying to say is I learned certain things, but other things I had to learn on the way.
That's all for this episode of the Biotech Startups podcast. We hope you enjoyed our conversation with Mark Kotter. Be sure to tune in for part 3 of our conversation to learn more about his journey. If you enjoyed this episode, please subscribe, leave us a review, and share it with your friends. Thanks for listening, and we look forward to having you join us again on the Biotech Startups podcast for part 3 of Mark's story. The Biotech Startups podcast is produced by Exceeder. Don't want to miss an episode? Search for the Biotech Startups podcast wherever you get your podcasts and click subscribe. Exceeder provides research labs with equipment leases on founder-friendly terms to support paths to exceptional outcomes. To learn more, visit our website www.exceeder.com. On behalf of the team here at Exceeder, thanks for listening. The Biotech Startups podcast provides general insights into the life science sector through the experiences of its guests. The use of information on this podcast or materials linked from the podcast is at the user's own risk. The views expressed by the participants are their own and are not the views of Exceeder or sponsors. No reference to any product, service, or company in the podcast is an endorsement by Exceeder or its guests.